Hey guys, welcome back. Don't forget to subscribe to our channel, the 10 Minute Tutor. This video is going to explain the process of DNA replication. If you need to understand the basics of DNA, check my other videos. The link should be right up here for you. To summarize, DNA is a double-stranded structure and it runs anti-parallel. Double-stranded, okay, and runs anti-parallel. These two words are very important to understand the process of DNA replication. So let me draw the structure of the basic structure of DNA. So let's say this is our two-stranded DNA. Okay, now this is a 5 prime and that's 3 prime. Okay, because it's anti-parallel, so this has to be 3 prime and this has to be 5 prime, right? Now in the process of DNA replication, what happens is these two strands, which are connected by hydrogen bonds, they get separated and then you end up like this. So this is your one strand, which is 5 prime was here, 3 prime was here, and here is your another strand, which is 3 prime here, 5 prime here. Okay, and then we make a new strand, which is right here and then right here. Now this is your 3 prime and this is your 5 prime, and this is your 3 prime right here, 5 prime right here. Now the reason I'm drawing that arrow is because DNA replication always happens from the 5 prime to 3 prime direction in the new strand, okay, also known as daughter strand. So this down, this is your parent strand, P-A-R-E-N-T, parent strand, and this is your daughter strand. And this process, this DNA replication, is known as semi-conservative. Okay, and as I mentioned earlier, it always runs from 5 prime to 3 prime direction. So the polymerization is always from 5 prime to 3 prime. Okay, that's important. Let's look at much more detailed structure of DNA. So let me draw double-stranded DNA. I'm gonna draw it with a green color. It's called origin of replication. So this is basically a sequence of DNA from where the polymerization actually starts. So let's say 5 prime, this is your 3, so this has to be 3 prime this side and this has to be 5 prime right there. Okay, and the replication starts from the origin of replication. But the problem with DNA is it cannot really start the replication unless you have something to initiate it, and that initiation happens if you have something called RNA primer. Okay, you need that primer so that DNA can just add onto that primer and then keep going. And that primer, RNA primer, is made by the enzyme known as primase. Primase makes that RNA primer. Okay, and then once you have that primer, you can start the DNA replication. So it runs like this. Okay, now here is your 5 prime and here is your 3 prime. Remember I said the DNA polymerization or replication always happens from 5 prime to 3 prime direction. The top one was easy. Okay, now the second one is a little bit tricky because 5 prime is here and you have this origin of replication right here. So technically, if you run from this direction like that, it's gonna break the rule, right? You're gonna have a 3 prime here and then 5 prime here, and it tells us it's running from 3 prime to 5 prime direction. That's not right. It always has to run from 5 prime to 3 prime direction when making a new strand. Move in a fraction. Okay, so technically we start from here like that. So this is your 5 prime, that's your 3 prime, again like that. So step by step, fraction by fraction, and then these fragments are called Okazaki fragments. Okay, this fragments strand is called lagging strand, L-A-G-G-I-N-G, lagging strand, and this continuous strand is called leading strand. Okay, you may be wondering how does this process actually start? Now we have this enzyme called helicase. This is our hydrogen bonds. Okay, this enzyme helicase actually comes at the origin of replication, and it starts unwinding this double helix. Okay, this is your helicase. It unwinds this double helix, basically breaks down the hydrogen bonds, and then so that the two strands are separated, so that the replication process can happen. Once this helicase separates the strand, we have a special proteins called single-stranded binding proteins. So they are right here. They are small proteins. They just sits right on the DNA strand. So once the helicase separates the strand, these single stranded binding proteins make sure that these strands are kept separated so that the replication process can happen. This polymerization from 5' to 3' direction polymerization, the attachment of these nucleotides are done by a special enzyme known as DNA polymerase 3. So that's the enzyme that basically creates a complementary strand, DNA polymerase 1. The job of the DNA polymerase is it removes this RNA primer and it replaces this primer with the appropriate nucleotides. Okay, and then now you may be wondering what happens to these small fragments right here, right? We need to connect these fragments, otherwise it's gonna create a gap. That fragments are connected by a special enzyme called DNA ligase. So DNA ligase basically connects, brings the nucleotides appropriate for this space and connects, so it becomes one long strand. Okay, that's pretty much it about DNA replication. That's a big thing to notice, is that the DNA, the origin of replication is a specific sequence in DNA recognized by the enzyme helicase. And then once you have that origin of replication, the primase makes a primer, and once you have that primer, the DNA polymerase 3 starts the initiation, and then complementary strands are being made. Okay, and then after that the other enzymes comes in and then play their respective roles. This is a summary of major enzymes and their functions used in DNA replication, and a clear picture of DNA.